This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 321. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, delighted to be with you.
Good to have you. From Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And from St. Louis, Missouri, Petra Levin.
Great to be here.
From your global travels, welcome back for doing a road trip recently. Today we have more amazing microbial science for you, and we're going to start with a snippet from Michelle. We're going to talk all about foodborne outbreaks.
Yeah, there have been a lot of outbreaks in the news, as I'm sure that our listeners appreciate. Most recently, the boar's head listeria outbreak. So I thought, and our paper today is going to feature food microbiology. So I thought I would start with something reassuring and really give a shout out to our colleagues at the CDC, who not only are keeping us safe from various infections, but also they've got amazing resources on their website for the general public, for healthcare practitioners, for public health safety experts. So I'm going to use the listeria outbreak as a guide to for our tour through the CDC webpage. So if you simply type in cdc.gov, you get really clear graphics and choices of what you want to learn more about. You can click, for example, on outbreaks, which is one of the headings at the top of the page. And when you do that right now, we see that there are 8 outbreaks featured. Many of them involve salmonella, but they're organized according to what is the source of the outbreak. So eggs, small turtles, bearded dragons, backyard poultry, all are spreading salmonella to people. And then we also have information on the measles outbreak, currently ongoing. Also, organic walnuts. There's an E. coli outbreak associated with those. And then also troubling is cinnamon applesauce pouches, which are often given to kids, are poisoned with lead and chromium. So if you click on whichever one of those you're losing sleep over, let's go with the listeria. Meats sliced at delis, the listeria outbreak. You're led to a page where they've got fast facts. Right now, there are 57 cases, 57 hospitalizations, 9 deaths, spread across 18 states. They let us know when they started the investigation, that it's still open, and yes, there has been a recall of the contaminated products. And then they give just a little blurb, including like very common, easy to understand language. Listeria is a hardy germ that can remain on surfaces like meat slicers and foods, even at refrigerator temperatures. It can also take up to 10 weeks for some people to have symptoms. Do not eat recalled deli meats. Some of the products have sell-by dates into October of 2024. So like a really brief, clear synopsis, and then you have the option of clicking, oh, I want to learn more. So you can click on one of their links. And for example, for the listeria outbreak, they will describe that more illnesses and deaths are being reported. Do not eat recalled deli meats. Again, some of them have sell-by dates into October. You can then click and learn more, the source of the outbreak, and they go into detail. Their epidemiological, laboratory, and traceback data show that meats sliced at the delis, including Boar's Head brand liverwurst, are contaminated and are making people sick. So those sold at a deli, especially either sliced or prepared at the deli, can be contaminated, and listeria easily spreads among deli equipment, surfaces, hands, and foods. And again, refrigeration does not kill listeria, But if you reheat to high enough temperatures before eating, it will kill any germs on these meats. So then, depending on what you click and what you're interested in, you can get more information. For example, they've got a photograph of a package of this boar's head ready-to-eat liverwurst products. They have the shelf life that is stated on the packaging, and it's labeled as Boar's Head Strausberger brand liverwurst made in Virginia. It comes in 3.5-pound loaves in plastic casing or various weight packages that can be sliced in the deli. And it has, again, sell-by dates from July to August of 2024 is the batch that's been contaminated. Okay, so I know what to look for. Check my fridge, pitch it if it's within these dates.
I have a
And
question
then,
for you.
yeah, go ahead.
Michelle. So they cut these meats on a slicer, and
Yeah.
can the slicer get contaminated and then spread listeria to other meats that were not being recalled, for example?
Yes. Yeah, I don't know what their practices are at this particular place, but if you were to do that experiment, yes, it can spread.
Maybe it depends on how much bacteria are contaminating the surfaces, right?
Yeah.
I'm just worried because, yeah, you cannot buy boar's head, but then if they slice something else next to it.
Right.
After someone just got a liverwurst.
Yeah.
Right.
But if you think you get out of this without not eating meat or deli meats, you're wrong. Because on the same CDC site, we learned that last November, there were listeria products linked to nectarines and plums that came prepackaged in the grocery store. So you didn't even have human beings touching the fruit at the store. It was already prepackaged, and you're buying beautiful nectarines and plums, and you're at the same risk. No slicing.
Contaminated at the source, basically.
Yes.
Yeah.
Right.
So listeria is everywhere.
I do think it's important to note that the levels of listeria are probably very important for infection, and also listeria is not equal opportunity infection.
Right. And we'll get into that using the CDC pages as a guide. So you can also click on another link and get the timeline, which is a formal term is the epidemic curve, where you've got number of people sick on the y-axis and the dates on the x-axis. So you can look to see what the intensity of this outbreak is. You can learn more about the infection. Tell us about listeria, their germs. People who eat contaminated foods can get sick. Infection is rare but serious. The good news is you can take steps to prevent infection. And then they have links, more information for everyone, the general public, for healthcare providers, for public health workers. If we go into each of those, you can click and learn more about symptoms, outbreaks, how it spreads, risk factors. Or if you're a healthcare worker, the clinical overview, clinical care and treatment. Or if you're in the public health field, what we do for surveillance, lab testing. Here's some publications that describe it. And then they have practical information, what you should do. And again, this is all really clearly laid out, very easy to absorb. And just the graphics are really impressive. So what you should do if you're generally healthy and not at higher risk of serious infection. Number one, don't eat recalled meats. Throw them away. Check your refrigerator. Do you have any of the recalled meats? Remember, listeria can grow on foods kept in the refrigerator. Oh, and then clean your refrigerator. Containers and surfaces that may have touched any of these recalled meats. And then if you're a person at higher risk, there's a different list of things you should do. And they point out that it's especially harmful if you're pregnant, 65 or older, or have a weakened immune system due to medical conditions or treatments. They can become seriously ill. So again, if you fall in this risk category, don't eat recalled meats. Don't eat any other deli meats you sliced at deli counters. You get sliced. Unless it's reheated to an internal temperature of 165 degrees or until steaming hot, then let it cool before you eat it. So that answers your question, Vincent.
Yeah. I like the map here telling what states have the most cases. I'm lucky in New York and New Jersey we have the most. Michigan seems free. South Carolina seems to have 1 to 2, and Missouri has 3 to 4.
Right.
So you can sort of get in the West. There's very little in the West, so it's all about distribution, right?
Probably.
Where it came from, and different plants make different products.
Yeah, so that's another really simple graphic that you can get to just quickly see what the story is with this health risk. They also have a section with what foods are linked to listeria, and they have a section about how listeria is spread: soft cheeses, raw milk, deli foods and prepared meats, pate and cold smoked fish, sprouts and melons. And if you click on any of the links, they go through the details and explain how each of those products can be contaminated and are in a special risk. And I can't leave it at that. Microbiologists have been studying listeria for a number of years, and I have linked in the show notes some work from Julie Terrio's lab, who years ago studied, when she was a postdoc with Dan Portnoy, how listeria can spread from cell to cell using the actin system, which is a cytoskeletal protein in our host cells. And it actually recruits actin to polymerize at the back end of the cell, and that projects the listeria, and it looks like it's swimming wildly. So you can watch these movies on Julie Terrio's webpage, see how they replicate in white blood cells that normally eat and kill bacteria, but listeria can escape from the vacuole, get into the cytoplasm, and then recruit the host actin and spread. So that's important, that mechanism, because that means they spread directly from one cell to another without coming in contact with antibodies or antibiotics or neutrophils, other white blood cells that normally eat and kill bacteria. And unfortunately, that cell-to-cell spreading also allows this bacterium to get into deeper tissues, whether it's the central nervous system of elderly people or the placenta and into the developing infant. So that's why pregnant women are need to be especially cautious of avoiding foods that may be contaminated with listeria.
Michelle, is there a long-term solution to this problem? Because it's always going to contaminate something, right?
Yeah, and that actually ties us into the paper that Michael is going to discuss. How can we do better surveillance? And I have also linked to a position piece by Stan Falco and Ron Atlas on the concept known as One Health, appreciating that the practices that we use in agriculture and farming and where we choose to live all changes our risk factors. So, for example, now that we build houses closer to the woods, people are more apt to be exposed to ticks that feed on deer, and maybe they carry Lyme disease. And then in the One Health concept that we'll talk about more with the food microbiology paper Michael will discuss, it is common practice in this country to add low levels of antibiotics to our animal feed. That allows the animals to grow more quickly, so you can get your cattle or your lambs or whatever to market more quickly, and it reduces the cost of our food. But obviously exposing the gut microbes to antibiotics, which they then poop out, and then if that manure is now used as a fertilizer for vegetables such as melons, if it's contaminated with bacteria and now the bacteria are antibiotic resistant, you can now introduce these antibiotic resistant bacteria into the food supply, into humans. So it's an example of this One Health, that when we change particular practices or policies, we can increase risk of nasty diseases. And that puts pressure on us, as you point out, Vincent, to come up with better diagnostics so that we can do environmental screening and predict where the problems are going to come and then prevent them, rather than wait until there's an outbreak and then working backwards to figure out the source.
All right.
So a question I had about the listeria was, what is the source of listeria? A lot of the illnesses, foodborne illnesses we get, are obviously fecal in origin, right. But listeria, I don't actually realize.
Soil.
I don't know.
Yep.
Soil. It's just soil. It's a common soil organism, and it comes in often on the animal hides. And so as they're effectively processing the animal, the listeria fall off. And if there's a growth medium, they'll effectively multiply. And what we found out from some of the statements that Boar's Head has made about this particular plant in Jarrett, Virginia, is that the plant was notoriously contaminated with insects and dripping water and thick layers of biofilm on apparatus. And so Boar's Head has elected to close this plant in the near term to see whether or not they could salvage it. And they ended up laying off workers.
Yeah.
So it's unfortunate that they failed the first principle of all food preparation, where cleanliness is the first principle. You keep everything in its most pristine state. And anytime you have insects in a food preparation area, you, of course, are bringing in those microbes.
And if you've cut some raw meat on one cutting board, you want to wash it with soap and water before you cut your salad vegetables that will not be
Michael,
cooked.
How would like nectarines and that sort of thing get contaminated? By contaminated water?
Contaminated water, they're washing them before they put them into the bag, and they're not diluting the water, or the water doesn't have a sufficient concentration of hypochlorous acid in it. They typically, remember when we did the story on ready-to-eat leafy vegetables like lettuce and spinach, they soak it in 50 part per million hypochlorous acid or bleach in order to kill off the bacteria that, and this was principally Salmonella and Listeria. And unfortunately, that strong oxidant then induces those microbes into a viable but non-culturable state. So even the tube and plates come back negative because they weren't able to grow. The bacteria were just sitting there effectively with their arms folded and say, okay, I'll grow when I want to. Thank you very much.
Michael, on a recent office hours, you said you could wash strawberries in some solution of vinegar so they don't get fuzzy. How much was that?
0.5%.
0.5% vinegar, because my strawberries always get fuzzy after a week.
Yeah.
I've also included in the show notes a number of reports from the American Academy of Microbiology related to use of antibiotics in agriculture, global food safety, how do we keep food safe from farm to table, and also pre-harvest food safety and security. So people are thinking about these problems, and the CDC does a great job of educating us. So if you are a teacher of microbiology or a member of the general public, and you know you're going to be meeting with family members or community members during an outbreak, you can easily go to the CDC website, get an up-to-date description of what the story is, and then share that information with your friends and family and colleagues.
Yeah, it's a very well done website. It's, people should take advantage of it, right? So thanks for bringing that to our attention, Michelle.
My pleasure.
They even have viruses there. Oh my gosh.
They do.
Looking at the oral Pouchet outbreak.
Yeah.
So
if you're losing
Mpox, they're all here.
Yeah, and Mpox.
If you're losing sleep, I recommend you go to the CDC website, and they'll at least make you feel like you understand, and it gives you some semblance of control, which always helps calm me down a little.
And just for your information, the disease has been renamed Mpox by WHO because they control disease names. But the virus names is controlled by the International Committee on the Taxonomy of Viruses, and it's still monkeypox virus. You're allowed to say monkeypox virus. Okay, that's the official name of the virus. But the CDC, in its ultimate political correctness, calls everything Mpox. You know, Mpox clade 1, and it should be monkeypox virus clade 1, but fine, I get it. It's a political
So this is like
well.
SARS-CoV-2 versus COVID?
Exactly
the disease is
so
Mpox.
WHO came up with the name COVID-19 and the ICTV SARS-CoV-2. And there's also HIV/AIDS. You know, there are lots of cases where you have the name of the virus and the disease, probably with bacteria as well, with microbes, right?
Well, this is why chickenpox, the virus, is very difficult for me to remember the name of the virus because...
Varicella zoster.
Exactly. But that name doesn't have anything to do with...
Because we have listeria and listerosis, right?
Right.
Is the disease.
That's easy.
Okay. Anyway, in the same theme, Michael has some interesting information about foodborne outbreaks?
A little troubling.
Michelle,
also a little troubling.
Well, this is the solution. And Michelle really teed up the problem statement well for us. The title of today's paper is Comprehensive Identification of Pathogenic Microbes and Antimicrobial Resistance Genes in Food Products using nanopore sequencing-based metagenomics. There are 20 authors on this paper, and that will take my entire time to read them all. So I'm just going to say this paper starts with Annie Wing-Tung Lee and ends with Gilman Kit Hang Su. And apologies to the other 18 authors. And they are from the Department of Health Technology and Informatics at Hong Kong Polytechnic University in the Hong Kong Special Administrative Region in China. And as Michelle brought us up to speed about the Listeria outbreak at the Boar's Head plant in Jarrett, Virginia, it will come as no surprise to routine listeners of TWiM that foodborne pathogens, particularly antibiotic resistance bacteria, or AMR bacteria, remain a significant global threat to all of us. However, there are limitations when using an approximate 150-year-old technology to evaluate the foods we eat. I mean, this is principally tube and plate technology that was pioneered by Koch and Pasteur well over 150 years ago. And they were simply asking the question whether or not to release a particular product. In the case of Pasteur, it was wines. In the case of Koch, it was beer. But today, a lot of what we're doing with food microbiology, it has to do with releasing a particular lot of hamburger tubes that we've all seen at the grocery store, where you can buy 2 or 5 pounds of ready processed hamburger meat, or luncheon meats, or even ready to eat greens. And what we learned from the CDC site just now, plums and nectarines. And so today, like all of the other disciplines concerned with assessing who's there, metagenomic sequencing of food products is surfacing as one of the premier methods for answering whether or not our food is safe, especially given the significant reduction in both cost and time to obtaining an answer. Now, consider the challenges of just releasing something as simple as ground beef from a meat packing plant, or shipping ready-to-eat spinach or salad greens to our local markets. The paper we're going to discuss today is going to use metagenomic methods in order to assess the presence of harmful bacteria and their containing antimicrobial resistance genes. And again, there are 20 people here, which will highlight the amount of effort that goes into actually carrying out this task.
And the scope of the project was really impressive.
Oh, yes, the scope. So they are employing nanopore long-read sequencing as their method of choice, for it offers an opportunity to provide more accurate bacterial taxonomic classification in comparison to short-read sequencing that we're all familiar with. So apologies to all who are familiar with nanopore sequencing, but we're going to have a short
There might be 3 or 4 of them out there, Michael.
To contrast the difference between nanopore sequencing and other methods that employ sequencing by synthesis approach, such as the one that's very popular amongst many of the papers that we have discussed, namely Illumina, and they typically refer to that as NGS, in order to answer the proverbial questions of who's there and how many are in your party. So what is nanopore sequencing? First and foremost, you can sequence RNA or DNA. It matters not. And like sequencing by synthesis, you need to add something to the nucleic acid you are seeking the sequence from, namely an adapter sequence, so you know the origin of the sample. And then in nanopore sequencing, you have to add a motor to the end of the DNA. And the motor is really the key, because it's effectively going to push the DNA molecule through the pore. And the motor is really nothing more than a glorified helicase that facilitates one strand of DNA migrating through the pore that was created in an artificially manufactured and electrically charged membrane. Unlike the other methods before, it does not require any amplification or labeling of the product. It's literally a direct approach to sequence the nucleic acid. So this offers an opportunity for it being faster, which is key when inspecting foods. And it can also sequence longer pieces of DNA, which also compresses the time to an answer because you're not constrained by the number of bioinformatic cores you can throw at it in order to reassemble your DNA to know who's there. And the best part, and I recall the bane of my postdoc years when saying this, high GC regions and then repeats in sequences. And any of you who've ever sequenced anything know GC rich regions and repeats are really torturous. But nanopore effectively just goes zippity doo-dah and pushes the stuff through. So how does it work? Well, here it's important to remember that membranes can be as tight as a molecule of hydrogen. So now imagine a tiny tunnel-like hole that's constructed in this hydrogen-tight membrane. That's the nanopore. Now visualize threading a long strand of DNA or RNA through this tiny hole. And as the nucleic acid passes through the pore, the different bases, whether they be A, T, G, C, or even a U, generate a different electrical signal in that membrane that is then transmitted through this energized membrane to a circuit where they are read as squiggle current. And you think I'm making up the word squiggle. Squiggle is actually the term that is used to describe the change in potential due to the presence of an A, C, G, or even U.
That's amazing. So that's the whole crux of this advance.
Yes.
It's literally current.
And they can effectively read up to 2 megabases, 2 million bases. That's just incredible.
It is incredible because in the old system, even the most modern kind, you're adding labeled bases. So you have to base there. So this is reading it without essentially directly off the DNA strand itself.
Yep. No manipulation.
No manipulation. It's not the
nothing
traditional methods, which were just adapted for high throughput. Yes, it's incredible.
So nanopore sequencing is reading these electrical sequences. And then the software, which is embedded on the chip, so you have now machine learning, is translating that back into sequence in real time. So, but if you really think about what's going on here, it's a bit like reading Morse code, another technology that's over 150 years old. But instead of dots and dashes, you're reading electrical signals generated by an A, T, G, C, or U. And most importantly, it can detect whether or not that base has been methylated.
Wow.
And recall that methylation is a heritable epigenetic marker, which often influences expression and does all sorts of other things. And in the case of bacteria, it tags it as self so that its own restriction enzyme doesn't trash the DNA to smithereens. So the key is that as the motor is unwinding the helix of the nucleic acid or the single-stranded RNA, it overcomes the charge inherent to the nucleic acid in order to push the thing through the pore, with the trick being each base has a different requirement to transmit and thus differences in the squiggle current. Now, like all sequencing technology, the limitation is it can't tell the ambiguity between living and dead microbes. However, the detection of non-viable cells and how many are resident in a product you are testing, like food, and whether or not they have antimicrobial resistance traits can still get the shoe leather epidemiologists on the scent to indicate previous contamination events of our food, triggering source tracking like Michelle gave us to the CDC, where shoe leather epidemiology actually protects us from many of these outbreaks. So if you will, this method fosters closing the barn door before the listeria leaves the factory. So in this manuscript, the authors assess the impact of food types and food attributes. So that means origin, retail price, and food processing methods on antimicrobial communities, and importantly, the antimicrobial resistance profile through this nanopore metagenomic sequencing. So here's what they did. We have 260 food samples collected from local markets in Hong Kong, both from the infamous wet markets and then traditional retail markets like grocery stores. So they had 51 samples that were from beef, 48 chickens, 81 pork samples, 23 sashimi samples. And recall that sashimi is raw fish, or in some cases, it can be raw meat sliced into thin pieces and often eaten with soy sauce. And then for the vegans of the world, 57 ready-to-eat vegetable samples. So I want to call your attention to their first figure. It describes the amount of work it takes to simply harvest the DNA from one of these samples that you have collected. And it's a really great graphic. You have the sample collection, and then they use something called a stomacher, which is really two paddles that effectively do what you do when you chew and grind, and they add some liquid to it that digests things, and then they were able to isolate their nucleic acid. Now, the authors offer that conventional culture-based approaches are time-consuming and have limited scope, and they're only able to detect less than 1% of bacterial diversity. And you got to know what kind of media to place things on. You have to hope you got the right growth condition, and God forbid they're anaerobes, and then you need all sorts of other special equipment. Now, there are a couple of things from the methods I want to call your attention to because they really do the methods well. The first is that they used a program from the R software package called index species to identify the bacterial species associated with different food types. And so that's INDIC species. And here they use the RG function that calculates the correlation coefficient R between each species and each food type, testing for statistical significance through a permutation test, where the values for a particular microbe, the INDI value, exceeding 0.25, which is nothing more computed for the association between the groups, were categorized as the signature species. So you're looking for a number bigger than 0.25. Another component you need to appreciate is the care with which they hunted for the antimicrobial resistance genes. Here, for the identification of antimicrobial resistant genes in the food metagenomic data, they used a selection process that specifically was looking for two criteria: an average read length quality score above 8 and a minimum length of 160 base pairs for the resistance gene identifier. These facts aren't really important unless you're in the field, but it really tells you the care and the rigor with which they went hunting for these antimicrobial resistance traits, as well as answering the proverbial question, who's there?
And Michael, I think your term hunting is really valuable here, because recall they started out with a sample of, a package of meat from a store or from a market.
Yes.
And they, digested it. They spun out the heavy stuff, tried to just get the microbes, but all that DNA would just be in one big gamish.
And they get to that in one of their figures because they show how much of the DNA is host DNA, yep, meaning the meat, and how much is microbial DNA, which is really freaking me out.
So the computational sorting is really important.
And that's often what takes the longest amount of time. The sequencing is typically fast, regardless of the method, but it's the assembly process, and that's the advantage in nanopore, because you get a much longer read, so you don't have as much to assemble. So this takes us to figure two, which offers us a snapshot of the microbial communities in the 5 food groups: beef, chicken, pork, sashimi, and the ready-to-eat vegetables. And that takes us, panel A is what really shows us what's actually going on in these food groups. When comparing the proportional abundances of the microbial groups in different food types, the archaea were found to be the most prevalent in the ready-to-eat vegetable category at 9.4% of the total microbial sequences, while bacteria were most abundant in chicken at 69%, and fungi and viruses dominated the raw fish and raw meats, whereas protozoa were the dominant group in pork. And if you grew up in my house, that's why my mother always cooked pork to death, because it was always well cooked, because she was making certain that none of those evil protozoans would get into her children.
So, Michael, let me clarify a bit. It was bacteria were the most common in all across the board. What they were saying is,
yes, I'm sorry.
Yeah, protozoa. What type of food has got the most protozoa? And it was pork. What type of food has the most archaea? Ready-to-eat vegetables.
Vegetables.
Yep.
Yes, I misspoke there.
And why the vegetables, I assume, have that because of the soil contamination?
Yes.
That's what I would think, yeah.
Yeah.
And they're not washing and scrubbing them to an inch of their life.
But I do think this is the figure where you were talking about what fraction of these reads came from host versus...
Right.
And that's figure 2B.
Correct.
And I think the part that this surprised me, because they're essentially taking up meat, and you would think that the bulk of it would be DNA from the meat. Do you want to explain? I mean, I can make some guesses as to why.
Well, they did that centrifugation. They did a centrifugation step.
Yeah.
So that the lighter bacteria could be separated from all the heavy gamish from the muscles of the animals.
So they're enriching. The centrifugation is enriching, but they're not able to remove all of the host DNA from it, because some of the stomacher is breaking up the meat fibers to smaller bits, and the nucleic acid extraction is indiscriminate. It just isolates DNA.
Right. And then I haven't thought about this in muscle tissue either. In animal muscle, since it's syncytial, what the fraction of DNA is in there at all anyways.
Yeah.
I don't know.
Yeah, I don't know the answer to that.
But I'd be feeling pretty good if almost 50% was microbial DNA.
Yeah.
Given the complexity of the sample they started with.
Yeah, no, I think so. I just was maybe surprised that you can do that from what looks like essentially solid meat, that you're still getting out so many microbes. Yeah.
And it's because of the washing step in the stomacher is not effectively digesting the meat down to its principal elements.
Right.
Like our intestines do. And the final panel from 2 is figure 2D, where the principal component analysis is figure 2D, where they were looking at the bacterial communities across the 5 food types. They exhibit similarities, except again for chicken and ready-to-eat vegetables, which showed overlapping patterns. And there was indeed a significant decrease in species richness as measured by both the Shannon and Simpson diversity indices. And here I'm going to do another short digression to offer the advantage. And again, this is tribute to the authors. Shannon diversity, its focus is it emphasized the evenness of species distribution. And in its calculation, it considers the abundance of each species and its relative frequency. And so in the interpretation then, higher values of the Shannon diversity index indicate greater diversity and evenness. The sensitivity, it's more sensitive to changes in rare species. So if you're looking for, say, for instance, Listeria in a cheese product, and you're going to expect lactobacilli, but that's the rare species. So the Shannon is better to look for our friend, the Listeria. Now, the Simpson Diversity Index primarily considers the dominance of species in the community. And this calculation then measures the probability that two randomly selected individuals belong to the same species. And this gets into the inside baseball of the molecular deconvoluting of the data.
Yeah, let's get into food risk, Michael.
All right. So by virtue of the fact that they gave us both types of information, this then provides valuable insight to us, the reader, into the biodiversity. And that takes us to figure 3. And here you get to see what's in your food. And you go, and they, again, have the 5 food groups in a beautifully colored figure. And in panel A, you see the 10 most abundant bacterial genera in the 5 food groups. And it goes from beef to chicken to pork, sashimi. And what really surprised me is that Pseudomonas was the #1 creature associated with beef. And I'm going, how is that getting into my beef? And it's probably through the butchering process. Similarly, it was in the pork as well. But then the other players that come up, chicken, on the other hand, its #1 microbe was Shigella. And Shigella is a bad actor because it can cause all sorts of uncharacterized, pneumonias of unknown origin. And until you get a molecular diagnostic or a growth diagnostic back from the lab, you won't really know it's Shigella.
Although at this point, we don't need to freak out because it could be dead Shigella, or it could be one of the avirulent, harmless Shigella. We don't know that it's a pathogenic Shigella species.
And that takes us to panel B, and which is why I digressed telling you about the indic values. And so you see beef, Carniobacterium divergens. Then there was an organism in beef that I never even heard of, Kurthia. How many of you guys have heard of Kurthia ever before?
I have.
Vincent? Vincent's heard of Kurthia. And Kurthia is a gram-positive non-spore former isolated, and this is probably why Vincent's heard of it, isolated from diarrhea samples associated with meats, milk, and soils. And you probably encountered it when you were looking at enteroviruses, is my bet. Panel C of figure 3 offers us the relative mean abundance of 15 potentially pathogenic bacteria in various food. And here's where we transition into looking for high-risk antimicrobial resistance genes in different food types.
Yeah. If I could back up to the Pseudomonas, I think one reason it may be so frequent is it's common in the soil, but it's also very versatile, can grow using a lot of different nutrient sources. So it's probably, if you've got it in your plant or in your harvesting equipment, it's probably hard to get rid of.
And again, Pseudomonas, one of the things I learned in microbial physiology once upon a time, it doesn't eat anything preformed. It makes everything from scratch. So it's really versatile at processing any carbon source you give to it into its base elements and then making more Pseudomonas from that. It's the ultimate recycler. They looked for the high-risk antimicrobial resistance genes or traits, including key beta-lactamase genes. They also looked for the glycopeptide genes. Those are things like resistance to vancomycin. They've also looked for the famous MEK-A gene from Staph aureus. And 123 out of the 260 food types that they sampled were found to carry, and this was, again, slapped me upside the head, 123 out of 260 were found to carry at least one high-risk antimicrobial resistance gene. And again, as indicated by their combined analysis with this risk group analysis that they have done, looking at the antimicrobial resistance algorithm that they used to deconvolute their data, which was, again, figure 4, principally panel C. Disturbingly to me, when analyzing each food type separately, the RGI analysis revealed that 21.7% of the sashimi samples and 45.6% of the ready-to-eat vegetables, and that tells you, ready-to-eat, you're not going to cook it, folks, had at least one high-risk antimicrobial resistance trait.
Yeah, so what do you think is happening there? What is it about our farming practices that expose it to more antimicrobial resistance genes?
It could be something as simple as using manure to fertilize the fields.
Or runoff.
And
runoff. Water used to irrigate.
And the CDC, in one of its other really great programs, Michelle, is actually monitoring streams and watersheds in the United States for antimicrobial resistance traits. And they're finding that pet waste that comes from municipal sewers also have antimicrobial resistance traits.
Wow.
So, the antibiotic resistance, because it's either adapt or die, is getting around, folks.
I mean, I think also what's interesting here, is that this technique, you could actually now start to survey whether different approaches, different farming methods or approaches actually, or different regions of the country lead to different outcomes. Because right now we just sort of have the spot thing and, wherever they sampled from, but you could actually screen, does organic farming or farming in the Midwest versus farming in, a place that might require, more low water
fertilizer, more water.
Yeah. Or
they did take one step in that direction, and they tell us in one of their paragraphs how many of the food samples came from China, how many came from Europe, how many came from North America. And I was heartened to see that the North American samples were on the low end.
1%.
Yeah.
That's in figure 5A, Michelle. And, bottom lining this incredible amount of information in this paper, and it's open access. So I encourage you to take a look at it. It's in Food, and it's really a great resource. In terms of overall occurrence, as Michelle was alluding to, Europe had the highest percentage at 7.7%, followed by China at 4.2%, and other Asia Pacific regions at 2.5%.
So are these for antibiotic resistance genes or bacterial contamination? The percentages that you just
5A. I got to see what 5A is. It may be bacteria or maybe virus.
It's bacteria. That's potentially pathogenic bacteria. Okay, so let's just take for a second though why Europe might have higher. So just saying, just a little thought exercise.
This is a sample bias. Could be sample bias,
right?
Because this is food markets in Hong Kong and wet markets in Hong Kong, as well as retail stores. So we don't know the distribution of wet versus retail and all of those other elements. And I didn't, unfortunately, have time to dig into the supplemental. Maybe it's broken out in the supplemental.
Right. But I think this is where you could do some hypothesis generating, right?
Yes.
So is it that when it gets to market, it gets contaminated, or is it contaminated before it gets to market, right? So that's a big question that I have. Another question is, maybe Europe has more of these because Europe is farming in Europe, most of Europe where farming occurs is super high density population, right? You don't have a lot of space. And I'm guessing Hong Kong, most of that is actually shipped from somewhere else. And so it's sort of like you can start to ask questions, where's it coming from? What are the fields like? And that, to me, is actually really exciting because that leads you to actually not only say what's there, but like interventions, exactly, which I think is really kind of cool.
To point out, foodborne pathogens such as Clostridium botulinum were commonly associated with food imports from China, 1.7%, and Europe, 1.4%, while Vibrio cholera was predominantly present in Europe. And I was scratching my head trying to remember the last cholera outbreak in Europe. But again, you have to have the phage in order to be virulent Vibrio. And so, we know there was a cholera pandemic in Europe, and it could just be still hanging around from the Middle Ages.
Right. Or there's cholera in the water, in Europe. And I think I've read a report, just in Scotland and places, there's non-virulent or a virulent cholera, right?
Or so, refugee camps, for example.
Well, then you have the virulent strains. So this is
water sources aren't separated.
Exactly, right. Are we talking about virulent or avirulent strains? Same for E. coli. Yeah.
Well, that's where the nanopore sequencing can come in, because you can look to see whether or not it has the phase.
Exactly.
This is all troubling, but there was one piece of good news that I would like to highlight. They looked.
Thank you, Michelle.
But they found that no food samples from China, North America, South America had any vancomycin resistance genes. And that's important because vancomycin is one of our, like, what's the term for it, Michael?
Last resort.
Last resort, top shelf. We don't use it unless it's resistant to everything else. So we're trying to protect it.
Yep.
But unfortunately, carbapenemase, which is another of those top shelf restricted antibiotic stewardship antibiotics, they also found a lot of that in the chicken. And that's associated with urinary tract infections. And in fact, 46.6% of food originating in the Asian Pacific region displayed high risk of antibiotic resistance genes. And again, only 16% of the food from South America exhibited high risk antibiotic resistance genes, in contrast to the other regions that only displayed a limited risk. And the carbapenem table, this was, figure 5B, it really gives you a snapshot of carbapenem, cephalosporins, which is a beta-lactam, like penicillin class of antimicrobial, vancomycin, and then the overall high-risk antibiotic resistance genes. And you can see it's broken down by food processing plants, retail places, and place of origin. And so, summing up, there are similarities in bacterial communities across all 5 food types, except for chicken and ready-to-eat vegetables, which display distinct profiles, particularly in terms of species diversity. Pseudomonas, as we already discussed, was identified as the predominant microbe across different food types. Now, previous research has suggested that Pseudomonas can spoil a broad spectrum of food products. And generally, I think that's what sours my milk in the refrigerator when I don't drink enough milk. But it doesn't pose a significant public health risk. But it's still, Pseudomonas aeruginosa is still a major contributor to hospital-acquired infections, which is indicative that it's going after the immune compromise. In the case of chicken, the most dominant genus was Shigella, which again can cause severe human infections such as bacteremia, where the bacteria are in your blood, or pneumonia. For sashimi, the prevalent genera were again Pseudomonas vibrio, Shigella, with Photobacterium damselae standing out. But Photobacterium damselae is commonly found in seafood, but it rarely causes an infection in people. But they also caution that during salmon processing, cross-contamination with Pseudomonas and Shigella occurs, raising concerns about the safety of the raw fish. In the case of ready-to-eat vegetables, apart from Pseudomonas, the most abundant were Rauhelena and Pantanea, which I have never heard of. And again, I'm assuming they're plant pathogens.
But I'm still washing my lettuce and my spinach.
Yes.
I don't care what it says on the outside of the package, ready to eat. I'm washing it.
I have another question about this. So this study was done all with sequencing, correct?
Yes.
So we have no actual data on what the numbers of these pathogens are. So just like you can have a PCR COVID test month after you've had COVID, or SARS-CoV-2 has infected you and still come up with DNA or PCR signal.
Well,
the question is, how do these numbers correlate, and how close are you getting to the actual infectious dose for some of these organisms?
Right. Well, funny you should mention that. In the show notes, I tossed into a paper that I tumbled into while I was looking at the literature. And there's lab on the chip, electrochemical biosensors for foodborne pathogen detection and review. And it's specifically table 1. And it gets to Petra's question exactly. It tells you how many Campylobacter, how many Shiga toxin producing E. coli you need, how many Salmonella you need to ingest. And you have the USDA standards. So, for example, Salmonella typhimurium, which we all know is associated with eggs, poultry, pork, beef, dairy products, seeds, herbs, sprouts, vegetables. You only need to eat 15 to 20 of those suckers. And what the meaning is, it's a potential health hazard and unfit for human consumption if you see greater than 15 to 20. But to get to your questions precisely about the concentration of nucleic acid, I guess if we can somehow figure out how to quantitate the amount of pathogen DNA, and I'm using a word where Mark Martin will yell at me for calling it a pathogen versus just microbial DNA, because
but also
Mark
there's
is
the
microbial centric.
The live dead issue.
Yeah.
So what we saw in this paper was the worst case scenario.
Yes.
I think in the end, you can't just go on DNA.
No.
If you have a sample that you suspect has whatever, you're going to have to culture it.
You're going to have to grow it.
And to the author's credit, they make that point in their discussion. They say, one of the next steps will be developing guidelines based on the numbers.
Right. What's nice about this, though, in contrast to PCR, is it's essentially a direct measurement.
Yes.
Yeah. So
no amplification.
No amplification
cheaper, faster.
Necessary. Cheaper, faster, but also for every sequence that they get out, that corresponds to one genome.
Yes.
Exactly.
Michael, did they find listeria anywhere?
No.
After we just talked about it for a half hour.
Well, the good news is Hong Kong, you don't have to worry about listeria.
Yeah, is that right?
Why not?
Well, no, you do.
All the food samples were from Hong Kong.
Yeah, all the food samples.
Purchased at Hong Kong.
But they weren't necessarily looking at the usual suspects that you would think. They weren't looking at processed cheeses, unpasteurized milk products, and other things.
Yep. Okay.
But this, and also in the show notes, I dropped 3 other papers if you're interested in how you look at food. And some of them are pretty cool, pushing the boundaries of detection. So if you're bored one evening, you can take a look at some of these papers. I try to find open source papers for you.
No reason to be bored in this world, Michael.
No.
No reason. But just turn on a TWiM or do something else. There's a
TWiP.
The world is just too interesting. Thank you, Michael.
Thank you.
So Michael mentioned show notes. You can go to microbe.tv/TWiM. And some of you may wonder, when we are talking about figure 1 and figure 3, we've started to put the figures in the YouTube version of TWiM. And so you can go there. I know many of you don't want to listen on YouTube. You listen on your podcast player, so you're not going to get that. But if you want the figures are in the YouTube. So youtube.com/microbetv.
That was an accommodation for our editors. They gave us the note.
Yeah, I understand. But a lot of people don't look at TWiM,
I know.
So
but if I used TWiM for teaching, and I would find that very useful.
No, the figures are great, but if you have an audio-only program,
right.
They're not great, and people turn off. They complain. I know. I get complaints all the time.
Yeah.
I try not to mention figures because in an audio podcast, which is what TWiM essentially is, you can't have visuals, right? Now, recently we're doing YouTubes with figures, so it's a little better, but it doesn't accommodate anyone. So I'm just trying to tell everyone what's going on here.
Thank you.
So microbe.tv/TWiM. If you enjoy these podcasts, we would like to have your financial support because we don't have any way of paying for this except your support. We don't do ads or any of that stuff. If you noticed here on YouTube, you can go to microbe.tv/contribute. We have a number of ways you can contribute, and Microbe TV is a 501c3 nonprofit entity in the U.S. Your contributions are federal, U.S. tax deductible. And finally, if you have questions or comments, we'd love to get them to twim@microbe.tv. That's our email address, twim@microbe.tv. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
My pleasure.
Michael Schmidt, Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
Petra Levins at Washington University in St. Louis. Thank you, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM, Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.